Okay, we're on page 36 of the note spiral, and so we'll be doing page 36 and 37. We'll be talking about a matched pair design. So I want to talk about a matched pair design before we look at the diagram. And in general, we previously talked about blocking. And blocking, you were controlling for outside variable that you believe things might impact the study, like gender. Well, you could sit there and go, you know what? I think gender impacts it. I also think age impacts it. I think body type might impact. I think where you live might impact it. I think economics may. And so pretty soon you keep dividing your sample down till you actually have only individuals. Well, that's not really very realistic, except maybe it is. The matched pair design is a special case of a blocking design. So the matched pair design is a special case of the blocking design where the individual is their own block. Now, in a matched pair design, the person gets tested twice. And they get tested twice because there's so many variations and every individual is different. So we basically control for all the variations within an individual. By the way, so here it is, to control for the variation within an individual or subject. We all are going to react somewhat differently because we're different. Our DNA, our genetic, our lifestyles are all slightly different. So that could all have an impact. So a matched pair design controls for all of that. Matter of fact, we should always run a matched pair design when possible, or we should consider running it when possible. And a matched pair design is extremely powerful. But to run a matched pair design, you have to receive both treatments, and that's not always plausible, so we can't always do it. So basically, how do you know if it's a matched pair design? Something is tested twice. To be a matched pair design, something is tested twice. In other words, it's receiving both treatments. All right, so why can't we always do this? Well, consider if you have taking a medication for a illness. If you go, hey, they're in this study, we're going to try this out on them for their cancer and see if this works. If it works, if treatment A works, then we'll never have an opportunity to assign treatment B to that person because they got well. So obviously they could not receive both treatments. And so it's not always plausible. I mean, if it's a weight loss program and the person lost the weight, you're not going to go back and say, hey, go regain the weight so we can try the other program. Now, the way the randomization works in this is one group of people will get treatment 1 first, followed by treatment 2, or maybe I should say treatment A, followed by treatment B, whereas the other group is going to get treatment B first, followed by treatment A. And at the end, we're going to compare results to see which group had a difference. Okay, so this is one of my favorite studies, and it actually walks through our questions, and it actually walks through 2 different types of designs. So 2005, Form B, question number 3. And by the way, the parts that are in red in the question, you are not going to be able to do until January or February, but the rest of the questions part of the question you can answer. So in search of a mosquito repellent that is safer than the ones that are currently on the market, scientists have developed a new compound that is rated as less toxic than the current compound, thus making a repellent that contains this new compound safer for human use. Science also believes that a repellent containing the new compound will be more effective than the ones that contain the current compound. To test the effectiveness of the new compound versus that of the current compound, scientists have randomly selected 100 people from the state. So we know we have 100 people, and up to 100 bins with an equal number of mosquitoes in each bin are available for use to the study. So we basically have 100 bins, so that would be essentially of mosquitoes, which is essentially 1 per person. After a compound is applied or the repellent is applied to a participant's forearm, the participant will insert his or her forearm into a bin for a minute, and the number of mosquito bites on the arm at the end of time will be determined. To me, this seems absolutely awful, although it's kind of humorous. And I have a feeling the way they get these people are they find people who need money, which is why they're always set up around colleges and universities because they know the university students are broke and need money. and are generally healthy, is for that matter too. So you've controlled for health by doing this. Suppose this study is to be conducted using a completely randomized design. So I see randomized design, makes me think place everything in a hat. So describe a randomization process. All right, place all 100 names of the participants in a hat and mix. The first group of 50 names will be assigned to the current compound. The remaining will have the new compound. The order didn't matter, just make sure you do that. The bins of mosquitoes will be numbered 1 to 100. Each participant will draw a number from the hat containing the numbers 1 to 100. The participant will then place their arm in the bin associated with that number for 1 minute. The number of bites for each treatment group will be compared to determine which compound worked the best. Please notice, we're comparing each treatment group. It's really important that you make sure you do that. The part that's highlighted in yellow, please don't worry about. We will look back at this problem in January or /February timeframe, and we'll talk about it then. The next one, suppose this study is to be conducted using a matched pair design. Describe a randomization process. So we need to talk about how we'd carry out a matched pair design. Remember, in a matched pair design, a person is tested twice. And you tested twice to control for the differences between the individual. So each participant will be randomly assigned to a bin as above. Since I've already talked about it above, I don't need to go through the 100 strips of paper. First 50 names drawn from a hat will receive the new repellent applied to the right arm and the current repellent applied to their left. The remaining 50 will have the new repellent applied to their left arm and the current applied to the right. So we switched arms for randomization purposes. Those who are assigned to an odd number will place their right arm in the bin first, while those assigned to an even number will place their left arm in first. Really important to swap which one goes first, because after the mosquitoes have fed on one arm, they may not want to feed on, they may not be hungry for the second arm, or maybe the first arm stirred them up and so they bite more of the second. We don't know, so randomization really important that you swap whether the new or current compound goes first. After 1 minute with the first arm, the second arm is placed in the bin. Then the difference in bites in each arm will be noted. In other words, we're going to compare the number of bites again to see what is more effective. Then it asked me which design is better and why. Well, why do we do a matched pair design? To control between difference in individuals. And I want you to think about mosquitoes and mosquito bites. I'm sure you've been out when the mosquitoes are biting, and maybe it was you or maybe it was a friend. But some people don't get bitten that much, and other people, they just get swarmed on. So basically, there are a difference in the number of mosquito bites that people get. So the matched pair design in part B is better than the completely randomized design because the matched pair design helps control for potential sources of variation from person to person, such as an individual's susceptibility to mosquito bites. All right. So basically, we talked about the individual. We're controlling for the individual, and we say that some individuals are more susceptible to mosquito bites than